Right, got it. And okay, thank you very much for the introduction, kind introduction. And I would like to thank organizers for giving me this opportunity to talk about our recent research results. My name is Shinpei Tanaka from Hiroshima University, and I would like to talk about our recent chemotactic, co-responsive chemotactic droplet systems. Okay, so there are several collaborators. Dr. Watanabe is from Hiroshima University, and Richard and Martin from University of Trento, and Professor Gorecki from Polish Academy of Sciences, and we are working together to study interesting behavior of droplets. And so let me give you a basic background about what we are doing. If you put a droplet on the surface of, say, water, and droplet has certain surface active substances, it can emit the surfactants around it, and if, well, of course, these surfactants can change the surface tension there, and if the surfactant distribution is symmetric, there is no motion, of course. But this symmetric surfactant distribution can be spontaneously broken. And once it is broken, the propulsion can feedback the inhomogeneous distribution around the droplet, so it goes on, it keeps moving. So let me show you this movie. This is a decanol droplet we are dealing with in this study, and you can see that at first the decanol molecules are spreading around the droplet, and it can show a certain amount of self-propulsion. Let me accelerate it a bit. So you can see the motion, right? But after a certain time, like this is, I think it's 7 hours after the initial motion, you can see no motion, because, well, even if there are decanol molecules around the droplet, this surface concentration distribution becomes symmetric and no symmetry breaking. But we can induce symmetry breaking as a gradient of chemical substances given as an environment, which is called chemotaxis, as you know well. And in this, under these circumstances, the droplet can follow the gradient of surface active, molecules. But what we are considering in this study is somewhat different from the usual chemotactic experiments. We would like to see the behavior of droplets if the gradient is given by two droplets. One is source of surface active molecules, and the other is sink of surface active molecules. So it's particle based, and because the sink, both of the droplets can move around, it's a co-responsive situation, and simple chemotaxis can induce interesting complex behaviors. That's what we expected. And there are previous researches, and let me show you one of them. This one is interesting because one droplet, this is a droplet inside the water, emits a certain attractant of a droplet. Oops. So this one emits attractants, and the other droplet follows the attractants. So it's a kind of run and chase behavior produced by this asymmetric relation between them. So we are creating a similar system, but on the surface instead of in water. So our source droplet is 1 decanol, it's a simple molecule which has surface activity. And the other zinc droplet is a paraffin. It's a simple liquid paraffin. And decanol droplet is dyed using Sudan black B. It's a dye, common dye. And what we observe is something like this. Okay, so this graph shows you the velocity against time. And when you have only decanol droplet, you can see the initial motion, but after that, it stops. So something like this one. So after this movie is after 7 hours, it stops completely. But if you add paraffin as a zinc droplet, you can see the velocity, well, they keep moving even after 15 hours, something like this one. So this blue one is decanol, and here I hope you can see the transparent paraffin droplet. And even after several hours or more than 10 hours, you can see this kind of interesting behavior between the two. Okay, so this graph is a magnification at T is 5, so around here. And the droplet, single decanol droplet cannot move, but droplet plus decanol plus paraffin can move, and its behavior is periodic. Okay, and we found that the concentration of dye, Sudan black B, affects the behavior very significantly. When the SB concentration is low, you can see something like this. This is a trajectory of two droplets. Orange one is paraffin, and blue one is decanol. So let me show you the movie. So when SB concentration is low, basically decanol droplet is rotating around the larger paraffin droplet, and it keeps rotating for a long time. And when the concentration of SB is high, this is decanol and this is paraffin, and they are slow, but if you accelerate it, you can see that basically decanol droplet is pushing the paraffin droplet. So it's a kind of run and chase behavior which was observed previously. But in the middle concentration, in between them, there's a interesting behavior. Actually, as you saw before. Sorry. Yeah. The decanol droplet is sometimes poking at the paraffin droplet, but immediately after the after close proximity, it is repelled by the paraffin droplet, but attracted again to the paraffin and rejected and attracted. And so it's a very interesting periodic behavior between the two. Okay, and this is a summary of the behavior. Y-axis is the distance between the two droplets, and the x-axis is the time of experiment. And when SB concentration is low, the distance is basically constant. Of course, you can see the significant deviation, but the binary star-like motion has a constant distance, so the distance is kept constant. And at high concentration, again, the distance is constant. But in the middle concentration, you can see the clear oscillation in distance. And this graph D shows the magnification at 5 hours, so around here. So you can see the oscillation, the distance oscillation. And of course, the binary star-like rotation has a slight distance oscillation as well, and it's more regular. But the oscillation seen in this middle concentration is not regular. We don't know how it happens, but it's an interesting complex behavior. Okay, so this is summary. When you increase the SB concentration, distance behavior is changing from constant distance oscillation and constant distance again. And we consider that this is caused by the surface activity of SB dye molecule. Of course, this is the dye, but it has a small surface activity, and this one is taken by Richard. The interface shell tension between decanol and water, and as a function of SB concentration, it's decreasing function. So SB has a surface activity, and this is paraffin water interface. And again, you can see the decreasing, it's a decreasing function as SB concentration. So this surface activity somehow causes this behavior change in the system. So this is it. We found that we can introduce the flow of surface active molecules from source to sink droplets, and this flow stabilizes the self-propulsion and as well as inducing very interesting complex behaviors. And I don't know how we can use this kind of phenomena to construct ALife, wet ALife, but we can use this kind of parts or a functionality to construct more, combining these functions, we can somehow construct more complex systems in the future. Okay, this is it. Thank you very much.
Thank you for a nice talk, Shinpei. We have some time for questions. If anyone has a question, please go ahead to raise your hand, or just, I mean, we're not that many people. You can just start talking if there's something. If there's no one else at the moment, then maybe you have a question, Jitka.
So you have shown the results just for water, and have you tried these experiments also for some surfactants? Or
no, we didn't add any surfactants to the system yet. Yeah, that's an interesting question. But if the water is saturated with 1 decanol, so it has it has saturated with surfactant, the droplet cannot move.
Yeah,
maybe
you have a stronger surfactant, then there's no gradient.
That's true. We didn't try it yet. Yeah, so that's interesting.
But it's really interesting, and I think we can discuss it later. Just in our droplet group.
Yeah, I would maybe also add a small addition to the part with the interfacial or the surface activity of the Sudan black. Maybe what is interesting to add there is also that when you measure the surface activity of Sudan black B on water, it seems to have no effect on the surface tension of water, but it only works in this way at the interfacial tension between water and oil phases, which is quite interesting to add.
Sorry, you did that measurement already?
Yeah.
Ah, good. Okay.
I did it with
Nakata. Black and black 4.
Yeah, I did it with
there are 2 dyes, and you tried.
That's interesting.
I remember the results.
Yeah, did it with Nakata-san.
Yeah.
Right. Any other questions?
I have one question. That's maybe a silly question, and forgive me for not knowing the literature, so this is a bit new for me. So I'd like to know, can you position this in regard to the literature? Like, what is new? What did you do that it was different?
Well, I haven't seen such a system so far.
Okay.
Well, you can induce the asymmetry between two droplets like this one, but basically the asymmetry induces the random chase relations. It's easy because one emits certain attractant, and the other follows the attractant. But what we would like to show here is that this kind of simple relation can induce much more complex behaviors, such as oscillations between the two droplets.
Okay.
So this is, well, this chemotaxis, well, the principle of chemotaxis is very easy, but you can use this easy principle to construct complex behaviors. That's what we want to study.
All right. Thank you.
Okay,
Thank you very much to the questions and to Shinpei. With that, we move on to our next speaker. That's Pasquale and Luisa. Pasquale is from University of Salento, and Luisa is from Milan. I don't know who of you will do the speaking, or you do a kind of duet.
We do both.
Oh, very nice.
The screen. I will read the first slide and then she will continue.
1 sec that I share the screen. Are you seeing my slides?
Okay. Yes. Okay. In this talk, we are going to discuss, recent advancement in synthetic artificial cell research, and we ask whether this impressive technical progress is leaving behind some important theoretical investigation. Of course, this is a collaboration between me and Luisa Damiano, who is a philosopher and epistemologist. Okay, next slide. So the interest in synthesizing and producing a protocell or artificial cell model started several decades or even centuries ago. And here I've reported, taken from the chapter written by Martin Ancic, a list of important contributions in this field. You can read important names like Alexander Oparin with the co-accelerate model, which was very important in the 50, 60, and so on. But maybe also the contribution of Fox with the proteinoids microspheres. But with the discovery of liposomes by Bangham, and then largely employed as a model by David Dimmer and Pierluigi Luisi, it was possible to create a model of cells which are much more, let's say, accurate and more similar in structure and in organization to biological cells. Next slide. And so this gave rise to the origin of the field of synthetic biology from the bottom up, where we try to assemble cell-like compartments that we call synthetic or artificial cells, putting together, for example, inside a liposome, DNA, enzymes, RNA, and so on, everything you need to create a kind of dynamics which is similar to some aspects of biological cells. You can do this by using modern molecules as in the drawing, but you can do also in primitive-like fashion, for example, using fatty acids or ribozymes or short peptides or whatever you can imagine which is prebiotically relevant. Interesting, you can also create artificial cells by completely artificial compounds like polymers, surfactants, peptide nucleic acid, inorganic catalyst, and so on. And also you can mix the things together in a variety of approaches, which is very rich and very promising for developing also many interesting technologies. Next slide. The history of this synthetic cell approach started more or less in the 90s, where the first examples of chemical autopoiesis was developed by Luigi Risi, and then developed in the 90s. In the few years, let's say from 1999 to 2004, a bunch of articles appear that reported the synthesis of proteins inside ribosomes, and this gives rise to a very rich research which continue until now with many reports of reactions reported inside vesicles and with very complex behavior that can be reconstructed. Next. Of course, the interest in these systems is that you can apply this kind of technology to create or to synthesize smart and programmable drug delivery agents. For example, already in 2018, the group of Avi Schroeder has published a system, a synthetic cell that synthesizes therapeutical proteins inside the tumors. And you can already imagine synthetic cells that can, for example, kill breast cancer cell or kill bacteria. Next. In order to make synthetic cells responsive to the environment, you need to develop a molecular communication module in the synthetic cell. This started the way of bidirectional or unidirectional communication between synthetic cells and other cells, and in general between synthetic cells and the environment. In the list, there are some experimental reports. One of the most elegant one is the one reported by Lettini from the group of Scher Mansi in Trento, who developed a bidirectional communication between synthetic cells and biological cells. And as reported in the drawing on the bottom right, you can imagine that a synthetic cell, the small object on the right, can feel a molecule released by a biological cell and then start an internal circuit to produce, for example, a molecule of interest that can, for example, be a toxin or a molecule that heal the cell. Next. Another example of recent development is the production of the synthetic cells that produce their own energy. At this aim, we have isolated chromatophores, which are small organelles from bacteria, from photosynthetic bacteria. And these organelles have all macromolecular machinery to transform light energy into chemical energy in the form of proton gradient, and then transform the proton gradient into ATP. We entrapped the chromatophores inside a larger vesicle, and in this way we demonstrated that it's possible to produce ATP inside a synthetic cell, and the ATP was used to produce, for example, RNA. Next. Okay, so this progress, of course, there are many others, gives rise to this question, whether these technical progress are leaving important theoretical investigation one step behind. And I would say that I and Luisa started this kind of investigation. I would say, Luisa, you can continue now.
So thank you very much, Pasquale. Yes, indeed, we think that there is a number of very interesting and important theoretical and epistemological issues that could be addressed by synthetic cell research, and anyhow are still neglected by this kind of research, despite the fact that they could really enhance the development of this research. And here we listed some of these issues, starting with the inheritance of the cybernetic movement for the future developments of synthetic cell research and its impact. The issue of the role of organizational theories of life in synthetic cell research, in the construction of synthetic cell research, and in the use of these synthetic cells, synthetic models of biological and cognitive processes. The issue of defining the complexity of natural and synthetic cells and building frameworks that allow us to rank different kinds of synthetic cells based on their complexity, but also the issue of the role of synthetic cells in cognitive sciences and explorative AI research, for example, the exploration of processes of emergence of minimal biological cognitive behaviors, minimal biological cognitive cells, minimal biological mind-like characteristics, and also the role that synthetic cells could play in applicative AI, and the role that information theories, and we are referring to syntactic information theories a la Shannon or semantic information theories a la McCake, a la Bateson, a la Nauta, could play in both these fields, so explorative and applicative AI. And indeed, all these issues are intertwined in the research path that with Pasquale we are constructing, at least since 2013, with Pasquale and with many other collaborators, depending on the specific segment of this path that we are considering the specific moment. Starting, as I said, with the issue of the inheritance of cybernetics for research on synthetic cells. For example, in this moment, we are focusing on the cybernetic foundations of the synthetic modeling of life and cognition, provided in particular by Rosenblatt, Wiener, and Bigelow in this very famous article that opened the cybernetic era, Behavior, Purpose, and Teleology, which was published in Philosophy of Science in 1943. In particular, we are working on 3 theses that we consider emerging from this article. The thesis according to which the biological body plays a critical role in cognitive processes, that is a contemporary thesis in AI, in the sense that emerged with the emergence of embodied AI in the 90s of the last centuries, and the embodied AI is the mainstream of the part of AI that is still modeling natural cognitive processes. The thesis that then, to synthetically model natural cognitive processes, we need to synthetically model the biological body, which embodied AI is doing right now through software and hardware models, that is computer simulation and robots. And mainly, and this is the thesis on which we are focusing today, to be scientifically relevant, synthetic models of the body should not simply reproduce biological behaviors, but should incorporate theories on biological organization, which, according to these authors, could be better done through wetware models. This was an idea that they exposed in 1943, and we are trying to implement today. And with their goal, which was the goal of overcoming proto-cybernetic standards in the synthetic modeling of life and cognition, which were standards converging on imitation, and trying to go beyond towards organizationally relevant models of cognitive and biological processes. So this line of research brought us to focus on organizational theories of life and to generate 2 main research lines related to this. A theoretical and technical research line asking how we can incorporate this kind of theories such as the theory of autopoiesis by Maturana and Varela, the chemodrome by Canti, the MR system by Rosen, how we can incorporate these theories in synthetic cells. And we are focusing on the use of the autopoietic theory in this case. And the second research line asking what are like organizationally relevant models of the cells, models that are incorporating these theories. And in particular, in a paper that we are writing in these days, we are asking if this model should be considered as the real thing, biological cells, or something else. And we are proposing the second of these options, and we are then asking what is the relevance of this kind of model for the scientific understanding of life and cognition. From the first of this research line emerged another research line related to the complexity of natural and synthetic cells. We wrote recently a paper about that, a short paper. And from the second of this research line emerged a research program in cognitive sciences and explorative AI that we called autopoietic synthetic biology artificial intelligence, autopoietic SBAI. And the explorative dimension of this program is try to position the synthetic study of embodied cognition, which is performed through software and hardware models by embodied AI, based in this case on techniques from synthetic biology at the fundamental level of inquiry about cognition, about biological cognition, which in our view is the level of minimal life. So the idea is that of attempting and constructing organizational relevant models that are wetware models of minimal cells based on the theory of our autopoiesis, the autopoietic view of the organization of life. And on this basis, trying to explore a bunch of issues. For example, the controversial thesis from the theory of autopoiesis, according to which life is cognition. So exploring the threshold of life, the threshold of cognition, their relations, but also studying basic biochemical mechanisms of signaling and regulation, which we consider fundamental for minimal forms of biological cognition in metabolism, in immune responses, and this also to offer the results of this research to research lines embody the eye that are studying higher level cognitive processes. On this basis, we are also developing programs in applicative autopoietic SBI, where the main goal is that of building wetware robots, SBI robots, for various applications related to health and environment, based on the idea that in complex contexts such as the biological body, natural environment, synthetic cells incorporating genetic circuits that are very efficient in highly controlled conditions would not be suitable. So the idea is that of creating instead distributed self-regulatory networks that in our view are expressed at the best through synthetic cells incorporating the theory of autopoiesis, the autopoietic theory of the biological organization, synthetic cells that are capable of perceiving and reacting autonomously to the environment in which they are embedded, which in our view would allow them to interact and perform much better than the synthetic cells I was describing before. And we're also studying, in particular Pasquale is developing very interesting research lines in these topics, the role of information theories in both these research programs, autopoietic SBI in its explorative and its applicative dimension. So these are the main research line we are developing at the moment. If you're interested in this, we invite you to our workshop, SBI workshop, which will be held on Friday morning. So thank you very much for your attention.
Yeah, thank you, Pasquale and Luisa, for a really nice and like thought-provoking talk. We don't really have time for any questions, but I recommend that if there's any questions, they can be put in the green room Slack, where we can, because I have some questions actually, because I find this theme very interesting. So we can continue the discussion there. In the meantime, then we have the next speaker now, that is Nathaniel Virgo from ELSI in Japan. And without further ado, please go ahead.
Okay. Hopefully you could see this.
Yes, you see everything.
Okay, good. So yeah, I'm Nathaniel Virgo. I'm at ELSI in Tokyo, and I'm going to try to make the case for a slow, messy start to the origin of life, where these terms slow and messy both have a relatively specific meaning in the context of this talk. And really making the case for this grand thing is quite a challenge in a short talk, so we'll see how well I do. So what I'm talking about here is the transition from chemistry to biology. So to set the stage, at some point, this planet, the Earth, about 4 billion years ago, or 4.4, whenever it was a very interesting non-equilibrium planet. It had liquid water, it presumably had lots of complex chemistry, it had an atmosphere, all sorts of stuff going on, but no life, no biology. And then at some point, at some unknown time after that, if you could go there and look at it, you would recognize that there was life there. It might not be the same as the life we have today, but how you would recognize it as evolving and metabolizing and doing everything that life does. So the question is, why did that process happen? How did it happen? Why is really the question. I'm less interested in the specifics. I'm much more interested in what can we say from a general perspective about why a planet would go from non-living to living. And I want to argue that this was a slow and messy process. And in particular, I want to argue that the chemical system that it began with was a slow and messy chemical system. So what do I mean by these terms? Well, by slow, I mean first that it probably took a long time. I mean, I guess that's just my opinion. But even if you think that life appeared instantaneously in the geological record, it still could have taken millions or tens of millions of years. Didn't have to. It could have been very quick, but it could have taken a long time, and I think it probably did. We know that evolution is slow, so why should the chemical processes that lead to it be fast and then suddenly have this slow anyway? Slow evolution. So, but what I really want to argue, the important thing is that chemistry on long timescales might be very different from chemistry on short timescales, and that not only is it different in character, but it's different in a way that's really important for the origin of life. So this is a talk about slow chemistry, really, but I can't say the things I need to say without mentioning messy chemistry, which is a term we came up with at ELSI that we use to describe chemical systems in which there are lots of reactions all going on simultaneously, producing all sorts of different products all at once. So in the lab, so I'm not a lab chemist by any means, but in the lab, there's a strong bias to because you care about yields, typically, not always, right? But there's a strong bias to work with easily controllable reactions, tightly controlled reactions that produce the thing you want to produce in high yields and good purity without too many side reactions and so on. Nature doesn't have the same bias. And biology perhaps does care about yields, but the chemical system before biology doesn't necessarily have the same bias. And there are lots of chemical systems that you can study that do have this messy property. The foremost reaction is one, produces lots of sugars. Hydrogen cyanide polymerization is another. There are the reactions that produce crude oil on Earth. So there exist these messy systems. And what I really want to argue is that when you have a system that's slow and messy at the same time, this is a really potentially very interesting type of chemical system for thinking about the why of the origin of life, why would protocells and so on emerge in the first place. So what I want to argue is that selection plays a crucial role in slow and messy chemistries, where selection here doesn't mean evolution by natural selection. It means selection, as chemists would use the term, where you can produce lots of complicated products, but you keep only some of them. Because they play some kind of functional role. And that's what we'll hopefully come to. So why would slow chemistry be any different from fast chemistry other than just being slower? Well, a way to think about this is to look at the Arrhenius equation, which says that for a typical type of chemical reaction, the reaction rate is proportional to e to the power of minus this thing called the activation energy divided by the temperature, with Boltzmann's constant in there as well. And what the activation energy is for a reaction to happen, first the reactions have all got to get there in the right place, but then they don't just instantly turn into the products. They first have to kind of configure themselves in just the right way, which kind of corresponds to going up this energy hill. And then only once you've got to the top can you go down the other side and produce the products. So a high activation energy basically means the activation energy is high. It kind of means things have got to happen in just the right way. These have got to come all together at like just the right speeds at just the right angles to get over this energy hill and then down the other side. So by slow chemistry, I mean systems where the activation energies are high or the temperature is low, which kind of is like more or less the same thing. I mean, it's not the same thing, but it's kind of equivalent because EA over T is the important quantity. So I don't mean that this constant proportionality is small. I mean that the activation energies are high. And the reason to think that this might be interesting is that as a sort of working hypothesis, I would guess that when activation energies are high, they're not just high, but they vary more among each other. Because if you've got lots of different reactions that all need something to happen just right for the reaction to happen. Like you wouldn't expect the thing that needs to be just right to be the same for all of them. So you'd expect them not just to have high activation energies, but to all have very different activation energies. So really, when I talk about slow chemistry, I'm talking about that type of system. And that type of system is interesting because the range of reaction rates is going to be exponentially huge when you're in that sort of situation. So some reactions will be vastly faster than others, and some will. So they'll all be slow, but some will be really, really slow. So here's a toy model that I mean, this is all the work in this talk was done years ago and isn't published. So you may have seen some of it before. So let's think about a toy model that can demonstrate why this should make a difference. So it's a very simple model, and I can explain it in just a couple of minutes. So let there be n different chemical species. They all have the same free energy of formation, so we can sort of ignore thermodynamic effects. What happens is the way the model is set up is that each species can catalyze the production of each other species from some feedstock that we just assume is there, right? It's always there. So each one catalyzes the production of another one, but the rates at which they do so are different. And the rates at which they do so are determined by the Arrhenius equation, the same equation as before, except that now the activation energies are chosen randomly for each pair of molecules, each pair of species. So they're not turning into each other. They're catalyzing each other's creation. So this is a non-equilibrium system and not an equilibrium one. So what we're going to do is we're going to take these kinetics. It's a non-equilibrium system. We're going to find the stationary state of that non-equilibrium system, but we're going to then vary this temperature parameter, which we can talk about varying the temperature, or we can talk about scaling all of the transition state energies. It's the same thing, right? Or like mathematically the same thing, physically a different thing. So we're going to change the temperature and plot how the steady state looks at different temperatures. So at a high temperature, or equivalently, kind of more or less the same, like generally the same rate constant, generally the same transition state energies, we see a uniform concentration profile. But as we decrease the temperature, what happens is that so if you would look at that uniform one, it would just be messy, right? You'd see a little bit of everything. Decrease the temperature, or equivalently increase all the activation barriers, then the system changes. So the mess is still there, but it's there at a very low concentration compared to just a few species. Now you might think, well, okay, so what, right? That happens in equilibrium systems too. You decrease the temperature, you tend to things tend to sort of freeze out, and you get this same sort of thing. But this is a kinetic effect. It's a non-equilibrium effect and not a thermodynamic one. So what's happening here, if you analyze this system, is that in the high temperature regime, everything's making a little bit of everything else. And so what happens is that you just get a little bit of everything. This is very similar, if you know it, to Stuart Kauffman's famous autocatalysis model, right? Everything makes everything else. You get this big autocatalytic set that consists of everything. But as you decrease the temperature, what happens is that the system finds the fastest cycle, which is not necessarily the smallest cycle. It's the cycle where A makes B, B makes C, C makes D, D makes A. It's the cycle like that where the product of all the reaction rates around the cycle is the fastest, right? And the system will then become dominated by that fastest cycle. So this is a form of selection, right? The ones that dominate the system are not the thermodynamically favored ones. They're these ones that have a function. They produce the next one in the chain, right? And if you think about autopoiesis, as mentioned in the last talk, right? I mean, this isn't autopoiesis, but there's something kind of autopoiesis-y about it, right? Everything making everything else in a big cycle. So yeah, this is what I just said. It's not because we're selecting for low free energies. It's a kinetic effect. Ones that are left are functional and they're selected, right? So in this slow, messy regime, selection becomes important. That was what I said at the beginning. So I've got a little bit of time, I think, so I can show you this slide. This is sort of different. This is an experiment that I did with Jim Cleaves. Did this a few years ago. It still isn't published, and that's because it's been incredibly difficult to analyze what's going on in this system, right? But what we did was we took a bunch of pre-basically plausible small molecules and a bunch of metal ions, a bunch of metal salts rather, and we mixed them all together in 200 combinations. So you're seeing 200 separate experiments here. We left them in a cupboard for about 3 or 4 years, I think. I mean, they're still in a cupboard, but the camera's not there anymore. This video shows, I think, about 1 year. The ones to watch are the ones kind of in the bottom half, because those are the ones with the metal salts. They have several different metal salts. So let's start the video. You can see they start to change color. Not so surprising, there's metals in there. But yeah, just watch it for a bit longer. There's a few weeks have gone by now, maybe a couple of months. The lights are going to go off and then we'll fix it. There it goes. Now keep watching. Did you see that? Some of them just suddenly went black after like 8 months. And then watch the ones just above those. They're going to do it. There you go. So yeah, there's things going on. There's chemistry happening over timescales of a year or so. Unfortunately, we don't really know what chemistry it is. I mean, we know what we put in the vials. We just don't really know what's going on at all. So it's kind of hard to publish it. But you know, stuff happens, and chemistry on long timescales might be different. So it's kind of exciting, and we'll manage to find a way to publish this at some point. So I've tried to argue that slow chemistry is important for the origin of life, because slow chemistry is fundamentally different from fast chemistry. And if you want to know the why of why you would get autopoiesis, or whatever you like to call it, autocatalysis, the origin of life, then maybe slow chemistry is the place to look, because that's where selection for function is what's driving the dynamics. So it's really an argument that that's an interesting thing to investigate. And if it's true, it kind of means we have to rethink the way we think about the origin of life. We often think of it as being this, like an environment where there's a high temperature and you get the right things in high concentrations all in the right place at the right time, and then the right reactions happen and you make these protocells with these lipids in this RNA, and then evolution takes over and it all kind of happens really fast. But maybe that's not how it happened at all. Maybe we need to look for a slow, cold, stable environment in which these slow, complex processes can kind of play out over a long period of time. And that would be a different way of looking at the problem. So that's what I wanted to say. Thank you very much.
Thank you, Nathaniel. This was really, really cool to see. And I really kind of agree with it to a certain degree, since we look at a lot of just physically dissipative systems that also oftentimes where you, let's say, the behavior emerges on longer timescales. As for example, the video I showed in the beginning, it looked like a lot was happening, but if you would, that was sped up a lot. If you would just look at it, it would just look like some bubbly water. And I find that really, really important to look at these. So that's what I wonder, if maybe some of these long timescales or these with these activation energies, that they maybe align with these physically dissipative rates, that there may be some correlation there.
What was that?
I just wonder if these the slow chemistry, and we have slow chemistry and we have slow physical dissipative processes. Maybe there's some kind of relation there, that these timescales that I don't know really how to formulate it.
I think so, definitely. I mean, like I used to play around a lot with like reaction diffusion systems and that kind of thing. I thought dissipative structures were really important. But like one huge difference between those and a cell is that a cell is a dissipative structure, definitely, but it has like these processes that take place across huge ranges of time scales. And that's like really different from the reaction diffusion spots and Bernard cells and so on. So I think that's actually a really important thing.
True. Very cool. Okay.
I have seen several times your video, but I'm wondering, it is like oscillation reaction when the color is changed, then it went back to the original color, or it's just now I have a link like this?
I don't think it's an oscillation. So what happens, I can show it again.
Yeah.
The ones to watch, I guess it's the fourth row from the bottom, and like vials 3 and 4 in the fourth row from the bottom. So at first they go orange, and then the orange starts to go pale, and then after that they'll turn black, and then they'll start to go pale again. But I don't think it's an oscillation. I think it's two different things.
Seems that
I think there's an orange thing comes. Sorry, go ahead, Jitka.
Yeah, it is like without any color, then it changes the color to orange, and then it went back to colorless, or
then it went back to colorless, and then it went black, and then it gradually became more pale. But I don't think it's an oscillation. I think the orange stuff is different from the black stuff, because there's a precipitate that appears when it turns black, and the orange doesn't precipitate.
All right. Yeah, I think we have to move on. I see that Chris has some question in the chat, but maybe Nathaniel can answer him there, or maybe on Slack. So with that, I think we start with the next speaker. That's Klára Pluková from Charles University in Prague. Hi, Klára.
Hi, yes, thank you. I'm gonna go ahead and share my screen. I guess you can see everything now, right? Thank you. Right, so I'm also going to talk about something related to the origins and evolution of life. I'm going to talk about proteins very specifically, and about proteins from reduced alphabets. So we're particularly interested in the transition from simple components on one side to scaffolds that are very useful today in biology on the other side, but also in sort of halfway through the process, we're interested how proteins could be useful halfway through this process. And I believe it has a lot of connections potentially to artificial life. So I hope I'm going to persuade you about this. And so today, proteins as we know them evolve mostly from pre-existing proteins by different mechanisms. But what I would like to talk about actually dates much farther back in history, basically takes us beyond the last universal common ancestor to times where the central dogma of biology was not yet fixed. So this really takes us to the roughly first 20% of the Earth's history. And most strikingly, different directions of research point to the amino acid alphabet of proteins not being the same as it is today. And it is assumed that the early alphabet was basically dictated to whatever was prebiotically available on the early Earth, which was a nice distribution of different amino acids, but interestingly, only half of those that we see today in the protein alphabet were present in this early set. Besides those, there was also a variety of different non-canonical amino acids, which for some reason didn't join the amino acid alphabet or were later purged from it. And the late amino acids, as we call them, were those amino acids added to the genetic code only later on when the biosynthetic pathways for their synthesis evolved. Okay, and so these transitions basically leave us with very fundamental questions. The first one being, okay, how could early proteins do the function and potentially the folding without these late amino acids? Or, could they do it actually? Could they work, or would they need some of these non-canonical amino acids to support the early cells? And I'm going to, I usually address these questions by two different approaches. One is bottom-up, the other is top-down, but I'm only going to show you the bottom-up, which is most related to ALife research. And I'm going to talk about sequence space that we created of these different alphabets to actually look at the intrinsic properties of these alphabets. And the model that we use is random sequences. The random sequences, we believe, are a great platform to study protein evolution because they're actually not biased by natural selection, by what we see in nature. And you could think that it's a very futile approach, but at the beginning of our research, we actually did a short study where we looked at some selected random sequences from the canonical alphabet. We looked at some of them one by one or whole libraries of them. And here is just a small window to what we saw. So this shows a very tiny selection of random sequences with high structural content and with low structural content. And we just looked at their expression soluble and insoluble in E. coli. And what we saw was, first, that these random sequences frequently express in E. coli, contrary to early beliefs of the community. We also observed that their average secondary structure content is very similar to biological proteins. And in fact, the less structure they have, the more soluble they are. Okay, so this was just something that we observed early on, but what we were ultimately interested in here were comparisons of different alphabets. And we created two vast libraries of protein sequences. One was from the full alphabet, and the other one was from the early amino acids, only those early amino acids that I just showed you some time ago. All these sequences were 100 amino acid in length, and we expressed them by self-re-expression system, producing about 10 to the power of 12 different variants in one tube. So we basically had two tubes of these libraries with this huge variety. Unfortunately, unlike Nathaniel's and Jim Cleave's shelf, these guys didn't have any colors. There was like nothing obviously going on and no funny chemistry. But it seems that there was more going on than we suspected. But what we actually wanted to know initially was, okay, how did these two tubes compare in their biophysical properties? How much is their behavior distinct? And there is not a lot of methodology that you can approach biophysical properties of these like large libraries. So what we did was, after expressing them in the self-re-expression system, we just separated the soluble and insoluble part by centrifugation, and then we exposed these fractions to proteolysis by two different proteases. Basically, assuming that the more compact structures would be more protected from proteolysis, and those that are more loose in solution, they would be cleaved by proteases. And that way we could distinguish these four different states, the degradable and undegradable parts of our soluble and insoluble fractions. And we did this in the absence and in the presence of chaperones. And okay, this looks maybe a little complicated, but what we were mostly interested in were these sort of darker blue areas, areas which represent the soluble and undegradable parts of the protein libraries, which is basically as close as we get to natural globular proteins, so proteins that are somehow compact and protected from proteolysis. And most strikingly, what we observed was that for the early alphabet library, we had a significant portion of this, something that we did not expect. On the other side, from the full alphabet proteins, this fraction was smaller, and generally the library is a lot less soluble. And this only improves after adding the chaperones. So after adding the chaperones, we get to similar level of structure occurrence in the library. But I just want to remind you that in this early alphabet, we don't have any aromatic amino acids. We don't have any positively charged amino acids. We don't have any sulfur-containing amino acids. So those guys that are usually used to, support structure in nature. So this was absent, and we didn't know, originally know why this was. It was a bit of a mystery. And I'm going to get back to this later on. But now I want to return to this perspective and say again that now we know that these early canonical amino acids can actually form something useful and something soluble. But what we still didn't know was, okay, what the role of these non-canonical amino acids could be. So to look into this window, we again constructed libraries. This time we were looking at peptide libraries, 25 mers that we synthesized chemically. And just to shorten it, I'm gonna show you only 4 of these libraries. The 10 early alphabet is very similar to what I've just been talking about. Then we have one where we added arginine as a representative of a canonical positive charge. And then we have 2 libraries which have some of the non-canonical yet prebiotically very abundant amino acids. And those amino acids are the typical biggest question marks. Why weren't they incorporated? They were really abundant. They on the first glance, they would make all the sense in the amino acid alphabet, but they were not. And we again looked at their solubility, and most strikingly, what we saw was that the canonical libraries are, reasonably soluble over the pH range, but those non-canonical are a lot more soluble over the whole range. So they induce solubility, but they actually decrease secondary structure potential. So the secondary structure potential here is represented by the CD signal as a function of trifluoroethanol titration, so increase in concentration, which usually induces alpha helical content. And that is true for the canonical libraries, but very untrue for the non-canonical libraries. So the biggest message here was that, okay, the early alphabet proteins can form structure and are significantly more soluble than the full alphabet proteins. But whenever we start adding some of these non-canonical amino acids, while we increase the solubility, we actually kill the secondary structure propensity. So this was our major observation from this bottom-up approach. I hope I have 1 minute more to just tell you that when we use the top-down approach, we actually learned that the way that these early alphabet proteins build the structure is by the support of small molecules, small organic and inorganic cofactors. So that was our mystery solved, but I'm not going to show you the data here. I just want to conclude specifically for this ALife community, our conclusion that the early alphabet that was used by during protein evolution is quite prone to secondary structure formation. It seems that it was selected for this property, and that folding and interaction of this alphabet can be assisted by cofactors. So I've seen a lot of talks about droplets, about vesicles, and I know that people are now thinking about incorporating peptides, amino acids, peptides, or small proteins. So my message here is, small alphabet as a really good model, I think, to start with for the solubility and even for its structure building propensity. And yeah, I just want to thank my group and our coworkers, and thank you very much for inviting me here.
Thank you, Clara. That was really cool. I think that's a good message to use these small alphabets. And I think I guess we can do one quick question from Pasquale. Or wait, was Chris? No, yeah. Pasquale, go ahead.
Yeah. Hi, Clara. Very nice talk. Thank you very much. We also worked more than 10 years ago about this topic, and we attempted to do 50 amino acid long never-bound proteins, random proteins, but the folding propensity was very low. And my question for you is whether you try also to make random protein shorter. And second question, whether you attempt also to predict the secondary structure based on the primary sequence.
Thank you. So I assume you're talking about random seed or never-bound proteins from the full alphabet, right? Yeah. Here, right? Yes. So yes, we also made them shorter. We also made 25 mers. The most significant problem that we encounter is the solubility. So these proteins or peptides are significantly and really significantly less soluble, which actually hampers most of the experiments. So we don't know why these early alphabets and all different kinds of combinations of early alphabets are so much easier to work with. We actually assume it's the level of complexity of the sequence space, that when we use a smaller alphabet, the complexity of the sequence space is a lot smaller. Also, I think the acidic nature always helps to solubilize. So this is the main trouble that we encounter. Predictions, we also made some secondary structure predictions, but only secondary structure. We still don't believe in the power of the tertiary structure prediction when it comes to completely unrelated sequences. Thank you.
right, great. So with that, I think we move on to Tomoko, who will give also a talk. Tomoko works with Clara and is also associated with LC, as far as I remember.
Yes. Can you see my slide?
Yes.
Okay. So thank you for giving me opportunity to speak in this wonderful session. I'm Tomoko Yamaguchi, belongs to the LC in Tokyo Institute of Technology, and now I'm living in Prague and doing the postdoc in biocell with Clara Loeffler group. So today I would like to talk about the in vitro experiment of the revolution of ribosome. When we're thinking about the life system, the central dogma is common feature for entire life system on Earth. It is the flow that DNA makes RNA and RNA makes protein. And especially RNA and protein are thought to be more ancient than DNA, and they existed from the beginning of the life. I'm especially interested in origin of life and how did the RNA and peptide emerged and have the functions. When thinking about the primitive stage of Earth, which comes first, RNA or peptide? So RNA can encode genetic information and have characteristic functions. So there is a RNA world hypothesis that RNA emerged first, and it is highly supported. However, the nucleotides are hard to synthesize prebiotically, and they are also prone to hydrolysis. Then how about the peptide? The amino acids are abundant in prebiotic world, and peptides are easily polymerized and much stable than RNA. However, the peptide itself does not have self-replication ability. Then we think that RNA and peptide could be co-evolved because RNA comes to be stable when it interacts with peptide, and also RNA peptide complex can have self-replication ability. So in biological system today, is there anything composed of RNA and protein or peptide? And the answer is yes, and it is ribosome, not liposome. So this is the macromolecular machinery that synthesizes proteins. This structure and sequence is highly conserved in 3 domains of life, which is archaea, bacteria, and eukaryote. Ribosome is the complex of protein and RNA and composed of 2 subunits, which one is the large subunit and the other is small subunit. Small subunits have decoding functions and each binds to the messenger RNA. The large subunit binds to the amino acylated transfer RNA and has catalytic functions. That means that those large subunits connect to amino acid together and make peptide. For these catalytic functions, the PTC regions, which represent the peptide transfer at the center, is very important. This leads to responsibility for peptide bond formations and also peptide release. Interestingly, this region composed of only RNA, and this structure and sequence is especially highly conserved. So it is thought that this part is most ancient part inside the ribosome. So there is so many studies about the evolution of the ribosome, and based on phylogenetic analysis, it is thought that ribosome has evolved by adding the extra segment of RNA and peptide and size up its structure from inside to outside. And Dr. Petrov and his colleague divided his evolution step by 6. And especially in phase 2, this large subunit is suggested to compose of only this PTC RNA, and it is also suggested that this RNA, only RNA, can show catalytic functions. Very recently, two different groups revealed that PTC ribosomal RNA only has catalytic activity. So in both groups, they synthesized the RNA and reacted with the substrate in the buffer conditions and measured the reaction product by mass spectrometry. So they revealed that catalytic functions of RNA, but if we added the peptide in this reaction mixture, does the RNA comes to be stable or show higher peptide transferase activity? If it's so, it also supports the evolution of the ribosome theory. So in our study, we focused on the next step of the ribosomal evolutions in phase 3. So this phase 3 stage, ribosomes start to interact with peptide. Within the 13 angstrom distance from the PTC center, Xiao et al. Revealed that there is 5 ribosomal proteins that interact with the RNA, this RNA. So we decided by using this complex, we want to detect if this complex shows catalytic functions. In order to check these catalytic functions with variety of peptide and conditions, we need high throughput method to detect this activity. And mass spectrometry is very strong method, but it is not easily accessed for everyone, and also it takes time to measuring. So we established another method that is fluorescent gel shift assay. First, we decided to use the reaction product as an indicator and mix this RNA and peptide with transfer RNA with fluorescent lysine and mini helix with puromycin. Mini helix is primordial of transfer RNA. So what is the puromycin? Puromycin is an antibiotic and mimicking the 3' end of trisyl transfer RNA. When this free amino group attacks this carbon, this amino acid bonds to the puromycin. However, in the next step, because there is no ester bond between ribose and amino acid, no additional peptide bond was formed and the synthesis will stop. If in these reactions, if peptidyl transfer reaction is occurred, the fluorescent labeled lysines transfer to the mini helices and mini helices start to fluorescent. After gel shift assay, we can detect the bond positions will be moved to the different positions. To confirm these reactions, we also prepared much smaller size of the mini helix so that this shorter mini helix shows the different fluorescent bond from the longer mini helix. Before starting our actual experiment, in order to check whether this method can work properly, first we use the full length of ribosome as a positive control. In this time, we used the E. coli 70S full length ribosome, and we didn't add any other proteins like elongation factors. And this is our result. So it is a little bit complicated, but the first lane is the the left gel is the signal from the fluorescence, and right gel is the signal from the RNA, which is dyed from by the cyber gold and green. In the first lane is the signal from the fluorescent lysine only, and lane 2 and 3 represents the only mini helix or mini helix, the smaller helix. And lane 4th and 5th represent the reaction mixture using the different size of mini helix. So as you can see here, in the positions of the ethylamine hierarchy, we can see the fluorescent signals. So it seems that this method is working, and peptide transfer reactions can be detected in these conditions, which means that I incubated this substrate with ribosome in 18 degrees or 37 degrees for 1 day. And those results are consistent with the previous report. In this previous report, they labeled the transfer RNA 1 mHz and reacted with the large subunit of the ribosome in the buffer conditions and check the reaction substrate by the thin layer chromatograph. In both substrate, transfer RNA and mHz, after incubated for a few days or few minutes, they detected the reaction substrate. So for further work, by adding those peptide with this ribosomal RNA and incubated with this substrate, we want to know whether this RNA peptide complex shows catalytic activity like ribosome or not. And also, if they show the catalytic functions, I also want to detect how does the existence of peptide affect the activity or the stability of the RNAs. So finally, I want to say thank you for all of the people who supported my research, and especially for the gel shift assay supported by Dr. Kosuke Fujishima and his student. And now the RNA peptide complex experiment is now underway with Dr. Kola Fohova and her super talented students. And thank you for my members, laboratory members, and all these fundings. Thank you for listening.
Thank you, Tomoko. That was a really nice talk. It looks like a lot of work. We would have time for one quick question. If there's one else, we can move on. But we can just see if someone has one question. Do I see any hands up? And if someone has any questions, then they can write them in the chat, and then Tomoko will be nice enough to sorry, or Slack, of course, yeah, to answer. Yeah, there's the to repeat this on the Slack. There's the green room Slack channel where we can do discussions for these workshops. So, so then thanks again, once again, Tomoko, and with that, I think I hand over to Julian Cartwright, who will give us a small introduction to a upcoming cost action.
Hello, thanks, Richard. You hear me?
Yes, I hear you.
Hi, Jitka. I see you lurking there in the background. So for the past several years, and I think starting from the last of these workshops, which was in Newcastle, right, in 2019, before the pandemic, I introduced what I was running at the time, which is just about to finish, which has been running for the last 4 years, which is called a cost action. And a cost action is a European money, not to do science, but to discuss science. So it doesn't give you money for research, it gives you money for conferences, for meetings, for lab exchanges, and so on. And it's a useful way to be able to form a community in science. So Jitka's been part of this. She's been running dissemination aspect, and several other of you have been part of this network of people. And for the past 4 years, we've been concentrating on something that we call chemobrionics, which so I can't share my screen until somehow you allow me to do that. So for the past 4 years, we've been concentrating on something called chemobrionics, which is based on very old chemistry, and at some point some of that old chemistry was mentioned in one of the talks, Leduc and Trauber and so on. These, where am I?
No, you can share, yeah.
These chemical gardens have been known for 4 centuries, but they turn out to be very relevant to the origin of life because they are what happens at hydrothermal vents on this planet and possibly on other planets. And so it's a very interesting way to look at both interesting chemistry. I'm a physicist, interesting physics, interesting origin of life stuff. And I should just add a plug that this is finishing now, but we have an upcoming meeting, the last annual meeting in Pisa in September. And although if you go to this page, it will say that the registration is now closed. If you are really interested in going, you can write to me or to Jitka and we can sort it out. So if you are interested in these things, you can come to Pisa and you will be paid if you're European. So that's finishing now. It's formed a nice community. At the same time, it's not everything to do with the origin of life or artificial life, etc. And several of the talks this morning have highlighted the importance of other aspects that come into the question of the origin of life. Now, the next point from that point of view is that for several years I've been, how do I do this? Trying to share the screen again. No, it's showing the wrong. Which one are you seeing? Are you seeing the same chemobrionics?
Royal Society Publishing.
Oh, okay, you are seeing the right one. Sorry. Yeah. So several years ago, with colleagues, since several years ago, we've been interested in information and DNA aspects of the origin of life. So several years ago, we wrote a special issue of philosophical transactions of the Royal Society. A lot of people contributed to that. It was really interesting. And so for several years, I've been interested in information and DNA together with my colleagues, Diego Gonzalez and Simone Giannerini. So we wrote another cost proposal, which has just got funded, and you are invited to participate in that. Let me share with you that. Where is it? Somewhere here. Here. So now do you see the cost one? Yeah. My screen gets confusing with too many things to see. So this, where is it now? The new cost proposal, which you can see on that screen, is about it's called ALife. It's about the integration between information theory and biology, and it's going to start in September for another 4 years. And you can sign up. So that on that page there is a description, a minimal description, and then at the bottom of the page it says action document memorandum of understanding. And if you go and read through that, it's 20 pages of a 20-page PDF file, which contains a basic sort of description of the field as we see it. If you're interested in the things that you read there, some of which have been highlighted in some of the talks today, about how we need to understand the evolution of the genetic code, about how we need to understand how life functioned before the last universal common ancestor, how you got from chemistry and also physics to biology. All of those things are part of this program of work. It's also quite nice because we, let me stop sharing. So this is going to, so there's going to be a meeting in Brussels to start the project in September, but the first scientific meeting is going to be next year, maybe in Bologna, where my colleagues Simone is, possibly somewhere else in Italy, possibly in Venice, where my colleague Diego is, but we will see. But probably March, April next year. That will be 2023. Now, that's quite nice, because this field, maybe one of the things it began with is this piece of work here by Schrödinger. Erwin Schrödinger was one of these people who was displaced by the Nazis. He ended up in 1943 in Dublin in the Institute of Advanced Studies before he left for the US. And while he was in Dublin, he gave a series of lectures on physics and biology, out of which came this book, What is Life?, which is required reading for anybody who's interested in these things. So that was 80 years ago, or next year. And then 10 years later, in 1953, two guys got together, an American biologist and a British physicist in Cambridge, against their department head's recommendation, and decided to crack the structure of DNA. They were told not to work on it, so they did. That's Crick and Watson, and of course they cracked it. They both wrote fantastic books about it. This is Watson's book. I recommend reading to any student. Crick's book, same. And that was 70 years ago, so next year. So there are some interesting anniversaries there. So we hope to have a strong start-off meeting. Some of you might have been to the start-off meeting for the chemobrionics network in Granada in 2019, and that was a nice meeting. So I hope that the 2023 meeting of this new ALife network will also be a strong meeting. And I hope it will meld together a community of people interested in coming from the theoretical side. One of the talks today mentioned the lack of theoretical developments. Well, I'm a theorist, I'm a mathematician and a physicist. I want to crack that side, but we also want to keep on board all the people doing wet stuff, whether you're chemists or biologists, and make a little community working on trying to push forward understanding information, life, dynamics of biological systems, etc., which of course will have relevance to artificial life, because as somebody else said in this little workshop, to understand artificial life, you should really understand the life we've got on this planet already and how that evolved. So you're all welcome. And you can ask me questions. Jitka is going to be involved in this one as well. So it's going to be good. And you're invited to get involved as well. If you're not European, we can't pay you always, but we can pay you at least once to come. So there is money.
Right, Julian, thank you. With that, because I have to move on to another room, I will thank everyone in this room. This was a really nice workshop. I hope we can repeat it soon. So thank to you all, and let's continue on Slack.
Yeah. Thank you.
Bye.